Right, hello guys. What we're going to do today is we're going to look at a Level 1 science paper, demonstrate the understanding of biological ideas relating to genetic variation. So we're looking at the 2014 paper. The best thing for you to do right now is to print out this exam, have it in front of you with a pen, so you can make notes while we're going over the content of the question. Then I will give you some time to answer the exam question and then we can mark it together. So the first question 1 is about genetics and the environment. 1A asks us to label the unlabeled bases A, G, C or T in the diagram of DNA shown below. And 1B asks us to explain the relationship between DNA, a gene and an allele. So before we tackle this question, we're going to go over complementary base pairing and DNA, genes and alleles. So DNA bases. The double helix ladder of the DNA molecule is held together by rungs, which are made of pairs of chemical cause bases. There are 4 types of bases, and they're usually identified by their initials A, C, G, and T. So complementary pairing occurs when 2 bases join together to form the rung of the ladder. So if you have a look at those bases up there, A, C, G and T, I want you to see if there's any way you could join those together. So just think about it for a moment. Right, hopefully you've seen that the shapes of the bases determine which ones they join to. So A and T join together and C and G join together. Now you have to remember this rule. If you're ever asked to join bases together, you have to remember that A and T join together and C and G join together. Right, the next thing we're going to go over is DNA, genes, and allele. So in the nucleus of a cell, you'll find chromosomes. Chromosomes are made up of long coiled molecules of DNA. Okay? The DNA is divided into regions which are called genes. The genes are sections of the DNA which give instructions on how to build specific proteins. For example, you have a gene for eye color. An allele is an alternative version of a gene containing different base pairings. So the way the bases are arranged determines the phenotype or physical appearance that you display. So the other thing to remember about alleles is that they can either be dominant or recessive. An example of a gene could be eye color. Alleles for the gene eye color could be blue eyes and green eyes. Right, so now we're back at the question. After going through that content, I want you to have a go now at answering question 1A and 1B. So if you just pause the video here while you answer the question. Okay, so let's go over the answers. Hopefully you got the A and T joined together, G and C, T and A, and C and G. 1B asks you to explain the relationship between DNA, a gene, and an allele. So hopefully you got an answer close to this. A gene and an allele are both made of DNA. A gene is a length of DNA that codes for a particular characteristic, whereas an allele is an alternative form of a gene containing a different base sequence. A gene for a characteristic contains 2 alleles. The dominant allele gives the phenotype of the gene. So remember that half your DNA comes from your mum and half your DNA comes from your dad. So every time there is going to be a dominant allele which shows the phenotype or the physical characteristic. Okay, the next question, 1C, asks us about variation within a species can be inheritable or non-inheritable. So 1CI asks us to give 2 examples of environmental factors that can lead to non-inheritable variation in plants. Okay, whenever it's asking you about environmental factors, it's asking you about things that are affecting something in its environment. So if you're thinking about a plant, its environment is going to consist of how much light it gets, how much water it gets, how much nutrients it's getting, what the pests are like, if there's any disease around it, and all of these factors are environmental, and that won't be passed on via its genetics. Okay, so just because a plant has the genetics to grow really tall, if it's not getting the right amount of light, the right amount of water, and the right amount of nutrients, then that won't be able to happen. But that's not to say that that plant cannot pass on the trait to grow tall. Okay, so the answers you're looking for there are light, wind, nutrients, water, anything environmental, pests, diseases. Okay, CII is showing us two pictures of the same species of plant. It's asking us to discuss how both inheritable and non-inheritable factors can result in the variation of these plants, and explain the importance of this variation within a large population of the plants growing in a changing environment. Because we're looking at both inheritable and non-inheritable factors, we need to be looking at the environment, meiosis, and why variation is important in plants in a changing environment. Right, so the first part to remember when we're talking about inheritable traits is that we need to remember that we get half our information from our mum and half our genetic information from our father. So we have to remember that that happens when the sperm and egg fertilise together to form a zygote. And when those cells divide by meiosis, they'll contain half the number of chromosomes. Okay, so a sperm and an egg contain one copy of each chromosome, half from the father, half from the mother. And the zygote formed contains 23 pairs of chromosomes, one half from the mother, one half from the father. Okay, so the stages of meiosis are also important. The first part, we've got the chromosomes and the nuclei. Okay, they're copied. One pair of each chromosomes are separated into two daughter nuclei. Now, the important thing that happens there is that they cross over. So crossing over is a process when homologous chromosomes pair up with each other and exchange different segments from their genetic material to form recombinant chromosomes. So what that means is that the chromosomes swap bits of information with each other so that you always are ending up with slightly different gametes. Okay, that's why sisters and brothers don't look exactly the same. So the daughters subdivide into 4 new gametes. So the two main stages of meiosis to remember is that the homologous chromosomes are separated and create 2 daughter nuclei. Homologous simply means that they have the genes for the same traits on them, so they're the same chromosomes. So there might be chromosome 1, chromosome 2, so those chromosomes are separated in half. The second thing to remember is that 2 daughter nuclei divide. The chromosomes themselves are separated and cross over, so that crossing over is what causes the variation, and then we get 4 different gametes. The next thing we need to think about when answering question 1CII is about variation. So I've got 2 pictures up on the screen now. One is showing a green background with orange and green bugs in the environment. So that's obviously meant to be mirroring a plant with 2 types of bugs growing on it. Now, in that environment, the green bugs are going to have an advantage because they're harder to be seen. So predators will be less likely to eat the green bugs. But there is also the orange colour bug in the environment. Now, if that environment was to change to maybe it's in an area of pollution and it's the leaf colours change, then the orange bugs will have an advantage. And that means that because of the variation within that species, they are more likely to survive if the conditions change. The next picture I've got up there is a picture of Darwin's finches. So Charles Darwin found 4 different types of finches living on 4 separate islands, Galapagos Islands. They all have different beak shapes, so that means that they are all able to eat different food. So the variation with those may have adapted over time by a natural selection, but it meant that they were able to survive best in their environment. So that is why variation is important. Next, I've got some sets of keywords, just to remind you again that you need to be using keywords throughout your answers in biology. So an allele is different forms of the same gene. We went over that earlier in this video. The next one is phenotype. So phenotype is the physical appearance of an organism. So whenever you're talking about how an organism looks, you should be using the word phenotype. So definitely in question 1CII, you want to be using the word phenotype when describing the differences between the plants. Another word you need to use is genotype. So that is the set of genes or alleles that an organism possesses. So that is its genetics, but its phenotype is what it physically displays. Homozygous is when two alleles are identical, and heterozygous are when the two alleles are different. So you won't need to use those two words in this 1CII, but you do need to be aware of them in biology. Right, so now we're back at the question. Hopefully you have enough information now to answer question 1CI and 1CII. Okay, so what we've got here is we've got the marking schedule for all of question 1. We're going to focus on question 1C, seeing as we haven't marked that already together. So to gain an achieved point for that question, you could have stated 2 environmental factors: water, light, nutrients, wind, disease, or pests. You could have stated that an inherited variation is due to genes, so it can be passed on, or that a non-inheritable trait is due to the environment and cannot be passed on. You could have linked an environmental factor to a characteristic that's been shown. So for example, if the plant's not getting enough water, it might not be able to grow tall, even though it's genetically able to grow tall. And you could have stated that variation is important in a changing environment. Okay, so to gain achieved in this question, you needed to have at least 3 of those points in the achieved column. Moving on to the merit column, just for question 1C, you needed to give an example of environmental factor and explain how it could affect the phenotype. So you needed to be using a keyword there to gain the merit mark. The second merit bullet point for question 1C is to explain how inheritable factors result in variation or survival. So you need to talk about meiosis, the mix of 2 parents, and how favourable traits are being passed on. So that's a natural selection point there. To gain excellence, 1CII was the excellence question in question 1. You need to discuss inheritable and non-inheritable factors, and how that results in phenotypes and characteristics. And you also needed to have the significance of variation in terms of survival of the plant population in changing environmental conditions. So once you've finished marking 1C, what you can do is you can go through and add up the bullet points you've got in the achieved column, add up the bullet points you've got in the merit column, and add up the bullet points you've got in the excellence column. And then you can look along the bottom of the mark schedule there, and you can see what your points equate to, whether that's achieved, merit, or excellence. Okay, so now we're moving on to question 2, which is about variations in humans. So the diagram below shows a relationship between gametes, zygotes, and chromosome number in humans. Okay, you can see that you've got cells in the testes, which is making sperm, to a zygote when it combines with the egg, and eventually forming an adult. Right, the first thing you need to do, question 2A, is to name the processes represented by A, which is above this arrow here, and B, which is when these two, the sperm and the egg, join together. Okay, so here you need to be talking about meiosis and mitosis. So remember, we've talked about meiosis earlier in this video. We talked about it being the ability to create sex cells, so sperm and egg cells, and that those cells have half the chromosome numbers of normal body cells. So a normal body cell is going to have 46 chromosomes. A sex cell or a gamete is going to have 23 chromosomes. The next thing you need to do is write the numbers of the chromosomes in the boxes. And then the last part of question 2 is to compare the chromosome number of the egg, sperm, zygote, and adult and explain any differences and similarities in the numbers. So we're going to go through, have another recap of meiosis quickly and mitosis. And then we're going to talk about chromosome numbers, and then we're going to come back and have a go at answering this question. Okay, to help us answer this question, I've given you a table here which shows the similarities and differences between meiosis and mitosis. So if we look at mitosis first, where does it occur? It occurs all over the body. So body cells is where mitosis occurs. It only divides one. It creates all cells apart from gametes. Those cells have 46 chromosomes in their nucleus. Mitosis is one cell separating into two cells, and the function of it is for cellular reproduction and growth. So all that means, cellular reproduction means that you've got, if you had a cut on your arm, mitosis would occur, new skin cells would be formed, and that would heal your cut. So all of those cells are going to have 46 chromosomes. Now meiosis occurs in sex cells only. It has two divisions, and remember, crossing over occurs to cause variation within the gametes. It creates gametes. They have 23 chromosomes. That is so when you have one sperm and one egg, when they fuse together to form a zygote, they have 46 chromosomes. It produces 4 cells, okay, and its function is sexual reproduction. Okay, so back to question 2. Hopefully you've been able to name the processes A and B. Process A is meiosis, and process B, when a sperm and an egg join together, is fertilization. The next thing you needed to do was to complete the diagram by adding the numbers of chromosomes in the boxes. So hopefully you got that the egg is going to have 23 chromosomes, same as the sperm. The zygote will have 46 chromosomes because the sperm and the egg have joined together. 23 plus 23 is 46. And then the adult will have 46 chromosomes as well. Okay, for question 2C, we're going to have a look at the marking schedule. So the answer here is that the egg and sperm both have the same number of chromosomes. This is so that when fertilization occurs, the chromosomes can exist as pairs. The chromosomes need to be in pairs so they can separate in meiosis. The zygote and the adult cells are both the same because every cell in the body needs to have an identical and full copy of the genetic information. The number of the chromosomes in the gametes is half the number in the body. Another thing you could add to this answer, seeing as you are talking about humans, is you could add in the numbers. So you could say sperm and eggs both have 23 and that the zygote has 46, same as the adult cells. Okay, question 2D is asking us about two brothers who have the same parents and are not identical twins. They will have different genotypes and phenotypes. Question 2DI and II are asking you to define the term genotype and phenotype. So I've given you the definitions earlier in this video, so hopefully you will be able to do that quite comfortably now. The next question asks you to explain how the two brothers with the same parents have different genotypes, and in your answer you need to mention the importance of meiosis and the role of fertilization. So in that question, you need to talk about half the chromosomes coming from the mother and half the chromosomes coming from the father. A good thing to mention there as well is in meiosis crossing over, so how the chromosomes are randomly assorted into the gametes, so you're not getting the exact same gamete being produced every time. So in order to explain that, you need to talk about meiosis and also fertilization. So you need to remember that 23 chromosomes come from the egg, 23 come from the sperm, they form the zygote which goes in the embryo. An important thing to remember is that this also happens in plants. You have an egg in a plant and pollen, and those two mixing of gametes creates variation in plants as well. Okay, so now we're going to go over the answer to question 2DI, II, and III. So for 2DI, you should have said that the genotype is a combination of alleles for each trait. and that the phenotype is a physical expression of a genotype, the alleles, the characteristic that's seen, for example, blue or brown eyes. Okay, defining genotype or phenotype gives you an achieved point. So again, it works the same. You go through, you look at the bullet points that you've got, you add those up as to whether you've achieved the question, gained merit in the question, or excellence in the question. So for question 2D, the brothers will have inherited half their chromosomes from their mother and half from their father. The actual chromosomes they inherited are due to chance because meiosis, which is the production of gametes, randomly mixes the chromosomes. Remember, that's crossing over, which results in a new mix of genetic information, and therefore different genotypes and phenotypes are displayed. Fertilization, which is when the sperm and egg join together, is also random, which creates more variation. So you don't know which sperm and which egg are going to join together, which means that the zygote has lots of chances of having different combinations of alleles and therefore variation. Okay, to gain a merit in this question, so you can look at the achieved one yourself. To gain merit, you needed to explain why gamete number is half the zygote number so that fertilization can occur. You need to explain that meiosis produces gametes which are different due to shuffling or independent assortment. Okay, you need to explain that it is random which egg and sperm join together, and you need to use inheritance to explain how different genotypes can arise as alleles in parents. To gain excellence, you have to get this point. Okay, to get merit, you need to have 1 point from merit for an M5 or 2 points from merit to get an M6. But to get an excellence in this question, you have to discuss how the two brothers could have different genotypes due to sexual reproduction, which produces a new mix of alleles, and because meiosis creates unique gametes, and in fertilization there is a random chance about which sperm and which egg join together. Okay, so now we're looking at question 3, animal breeding. So an animal breeder wants to produce sheep with white wool, but sometimes white sheep produce lambs that have black wool. Animal breeders often use one male sheep to mate with all their female sheep. So question 3a asks us to give all the possible genotypes for each phenotype. Remember that genotypes are how the alleles are arranged genetically, and the phenotype is how the physical appearance is displayed. So the capital A is representing the dominant allele for white wool, and the lowercase a represents the recessive allele for black wool. So hopefully you would have worked out that there's 2 possible combinations for white wool and 1 combination only for black wool, because whenever there is a dominant allele present, that dominant trait will always show. So for a recessive allele to show up, it has to be 2 recessive alleles. So hopefully you've worked out that white wool can be capital A, or capital A, lowercase a, and black wool can only be 2 lowercase a's. Okay, 3b is a long answer question. It asks you to discuss how a farmer could develop a group of sheep that are pure breeding for white wool. That means that they only ever produce white wool. In your answer, you need to state the genotypes of male and female sheep the farmer should use to breed from. You need to explain how the animal breeder can determine the genotypes of the male and female to produce sheep that have all white wool. And you need to use Punnett squares to show that, at least 2. And then you need to explain how the animal breeder could make sure the offspring will always be pure breeding. Okay, so the first thing that you need to know is that heterozygous is a dominant and a recessive allele. Okay, and that one will always be white, because whenever the capital letter is there, the dominant allele is there, that allele will always show. Homozygous dominant is 2 capital A's, and that will be white as well. And homozygous recessive is 2 lowercase a's, and that is the only time that a black sheep will be displayed. So the first thing you need to do is you need to go through and do your Punnett squares. So the first Punnett square I'm doing is between a homozygous dominant sheep and a black sheep. The reason that I'm going to cross these 2 together is to see what the offsprings are. I can test to see if the sheep is actually homozygous dominant. So you can see there that all of the offspring are white, so that shows that there is a strong likelihood that the sheep is homozygous dominant. The second cross I'm going to do is between a heterozygous sheep and a black sheep. So you can see if you've got a heterozygous sheep, there is a 50-50 chance that the sheep will either be white or black. So this cross needs to be done more than once to remove the chance that the white sheep's heterozygous. So you need to do this cross heaps of times to make sure that it is actually a dominant sheep. So as soon as a black sheep appears, the farmer would know that the sheep was heterozygous. So hopefully this is enough information here for you to go on and answer this question. So have a go at answering it, and then we will check with the mark scheme. Okay, so question 4 is asking us about tongue rollers and non-tongue rollers. So we've got a family tree below. People who are tongue rollers are displayed as that picture there, and while those who cannot roll their tongue are a smiley face. So you need to use the family tree to work out the genotype of individual 5. So this is individual 5 here. They've got a smiley face, so they are not a tongue roller. Now, important things to see is that we are told that tongue rolling is dominant and non-tongue rolling is recessive. So if person number 5 here cannot roll their tongue, then they have to be homozygous recessive. The reason they have to be homozygous recessive is if a dominant allele was present, then individual 5 would be a tongue roller. So therefore, they must have both recessive alleles present. Okay, so question 4b asks us to use the family tree to explain why individual 6 must be heterozygous. So the first thing we need to do is identify which one individual 6 is. This is individual 6 here, and they can roll their tongue. So individual 6 is a tongue roller, so they must have at least one dominant allele present for the tongue rolling to be expressed. So they have to be either heterozygous or homozygous dominant. The next thing we need to do is have a look at the offspring that person 6 has produced. So we can see here that neither of those offspring can roll their tongue. So both of 6's parents are non-tongue rollers, which means that they have both recessive alleles. So they have to be homozygous recessive. We know we get one allele from each parent, so I want you to use this information to answer the question. Remember that individual 5 cannot roll their tongue, so that means that they must have homozygous recessive or 2 lowercase t's. Right, question 4c, the last question in this paper. You need to explain why genotypes for individuals 3 and 4 must be heterozygous. So you need to draw at least 2 Punnett squares. You need to explain why they cannot be homozygous dominant or homozygous recessive. So these are 3 and 4 here. They can both roll their tongue. 2 of their offsprings can roll their tongue, but individual 8 cannot. So we need to focus on individual 8, because that must mean that that individual has got recessive alleles from somewhere. So if we cross 2 heterozygous people together, we get a 75% chance of being able to roll your tongue and a 25% chance of not being able to roll your tongue. So that accounts for individual 8. If we cross a homozygous dominant with a heterozygous, we get a 100% chance that the offspring can roll their tongue, which doesn't fit with individual 8. So individuals 3 and 4 cannot be homozygous recessive, as are both tongue rollers. For them to be tongue rollers, each of them must have at least one dominant allele. It isn't possible for them to be homozygous dominant, as one of the offspring is a non-tongue roller. So this second Punnett square here proves that they cannot be homozygous dominant. Child 8 is homozygous recessive, so the parents must have a recessive allele, because they both can tongue roll, they must also have a dominant allele. So they can only be heterozygous.